Hello and welcome dear viewers to the another informative video on discriminatory and clinically relevant dissolution testing and dissolution specifications. This video will discuss regarding these two dissolutions based on the example of capsule formulation, and the capsule formulation is of soft gelatin capsule formulation. Discriminatory dissolution means the capacity or capability of the dissolution method to distinguish the significant changes in the composition and manufacturing process of the formulation. And that's why, due to this capacity of the dissolution method, the discriminatory dissolution method can catch the good batch and bad or mismanufactured batch. Dissolution method which passes all the batches may be called as under discriminatory or permissive, or the dissolution method without any discriminatory power, and the dissolution method which doesn't pass any of the batch, that is called as over discriminatory method. So the method should not be under discriminatory or over discriminatory. Dissolution method discrimination capacity depends on the dissolution method development. If not developed properly, the dissolution testing conditions are not proper and the dissolution specifications will be permissive. In the regulatory language, the dissolution specifications may be permissive or too liberal or under discriminatory. For example, too wide specification like 75% in the 45 minutes for a dissolution of 100% in 15 minutes. So if your bio batch is having 100% dissolution release or drug release in 15 minutes, and if the dissolution specification are given like 75% Q in 45 minutes, then that specification or that dissolution method is not having any discriminatory power, and that method will pass all the batches. Then too narrow, like 80% in 45 minutes for dissolution of 80% in 45 minutes. That means for this type of specification, the batches will be required to be tested at S2 and S3, and also they may fail because it is very cut to cut type of specification and which needs to be relaxed little bit. Then coming to the discriminatory power, this is the ability of the discriminatory dissolution to distinguish the good batches and the bad batches. Good batches are those batches which are equivalent to the clinical batch or bio batch and which are supposed to be similar to the bio batch, and the bad batches are those which are not similar to the bio batches. So discriminatory power is the ability of the discriminatory dissolution method to distinguish the changes in the quantitative composition of the formulation, material specifications, or using the slightly modified process parameters. The batches with meaningful changes are compared with the batch manufacturers with finalized composition and process. Change in the drug substance particle size, crystalline amorphous form, binder grade like HPMC E5 to HPMC E15, quantitative formulation changes like tablet binder, descending lubricant level change by 10% to 20% compared to the finalized composition, and slightly modified process parameters means, for example, weight granulation, higher kneading time, higher solvent uptake. For direct compression, it will be higher lubrication time, higher hardness of the finalized tablet than the optimized ranges, or higher tamping to the powder filled capsule. Then coming to the clinically relevant specification. Till now we have talked regarding the discriminatory dissolution method and discriminatory dissolution specification, but now it is clinically relevant specification, and nowadays the regulatory authorities are going towards the clinically relevant specification. Why? Because for the formulations which are given orally, the dissolution reflects into the clinical action. That's why these clinically relevant specifications are required. So these are given on the basis of critical bioavailability attributes. These are called as CBAs. These are the formulation or process attributes. Those are expected to critically impact the bioavailability of the drug product. And bioavailability means the absorption rate and the extent. So absorption rate means Cmax and extent means AUC. So these parameters may impact the Cmax and AUC. So these parameters are called as CBAs. Critical bioavailability attributes to be clearly identified, detected and controlled. Also, IVC or IVIV are to be established with physiologically based pharmaceutics model, biopharmaceutics model, that is called as PBBM, and or any suitable in vitro in vivo data should be there to have the understanding of the CBAs. The specification which takes into account the consideration of clinical effect of the variations in dissolution ensuring a consistent safety and efficacy profile is called as the clinically relevant specification. That means your dissolution specification is directly relating to the action of the drug. So in vitro and in vivo action is correlated and on which the clinical relevance is justified. So the reference for this is clinically relevant dissolution specifications, a biopharmaceutics risk-based approach, a FDA perspective. One publicly available document is there. From there you can understand the other things as well. So based on the BCS also these CBAs can be understood. Then based on the risk level of the formulation and the BCS classification of drug substance, the risk levels are given, which involves very low risk, low, medium, high and very high risk. So when the risk will be low or very low, if the drug substance is BCS class 1 or 3 and if it is rapidly or very rapidly dissolving and standard dissolution test conditions are there considering the high solubility, then it will have a very low risk. Then low risk, dissolution test with scientifically sound condition. Limited method development is needed to justify the method and or acceptance criteria. Here also the high solubility or high permeability can be taken into consideration. Then coming to the medium risk, in vitro approach to mitigate the risk. Dissolution test should be target to detect the meaningful changes in the identified CBAs and to provide the insight into in vivo performance. So from here the clinically relevant specification starts. Here all the CBAs are required to be identified and controlled with the specific discriminatory and clinically relevant dissolution specification. Then coming to the high risk, here IVIVC and IVIVR is required to support the patient-centric dissolution test based on the available in vivo data or PBPM. So PBBM is required, IVIVC is required to have the clinically relevant and patient-centric dissolution test and specification. Then the risk is very high. That time in vivo studies are used to develop IVIVC or IVIVR to support the patient-centric dissolution test. So these are the risk levels and these approaches are used for the risk level involved in the formulation. Then the example of soft gelatin capsule formulation I have taken to get the in-depth understanding of the clinically relevant specification and the discriminatory dissolution specification. So these two things are related and these are not separate things. So from discriminatory dissolution we are going to the clinically relevant specification. See here the example is hypothetical and from this hypothetical example we will understand the relevance of the formulation, drug substance, process, specification and the clinical output. So drug substance physicochemical properties is BCS class 2. So drug is BCS class 2 which is low soluble and high permeable. Solid state form is crystalline form and crystalline form is insoluble throughout the pH. Solubility practically insoluble in physiological pH range. So the molecule is practically insoluble. It is not having any solubility. Drug substance biological properties. The Tmax is approximately 1 hour if the formulation is prepared for soft gelatin capsule and the drug substance is dissolved, then that formulation is showing a Tmax of approximately 1 hour. This justifies the BCS class 2. That means the drug is insoluble but high permeable, and due to this high permeability the Tmax is coming to around 1 hour. Permeability is high, thus the dissolution becomes critical rate limiting step. And for dissolution, the solubility will become the rate limiting step. Now for solubility, the crystalline form or soluble or dissolved form of the drug substance into the formulation will become the critical stage. Then formulation properties. The formulation developed for this type of formulation is liquid filled soft gelatin capsule. Why? Because the API is insoluble over the pH range, and based on the physicochemical properties, it has shown good solubility into the oils or medium chain triglycerides and the non-ionic water in oil soluble surfactant as a solubilizer. So these type of materials tried and API got dissolved into non-ionic oil in water surfactant. So that used as a solubilizer, that API dissolved into the solubilizer and filled into the soft gelatin capsule. Soft gelatin capsule formulation selected based on the API properties and to have rapid API release and API to be in the solution form in the GIT, so that the permeability will be there and the API will get absorbed at a faster rate. Then coming to the soft gelatin capsule formulations, CBAs, critical bioavailability attributes for formulation. This is cross-linking of gelatin shell. If cross-linking is there, then it will lower the rate of dissolution. It will give a variable result into the dissolution. Then crystalline insoluble API in capsule. If the crystalline API is present into the capsule, then the dissolution will get hampered. Crystalline API is insoluble, and this crystalline API is only soluble in the surfactant used or base used as a liquid for the soft gelatin capsule. And if this API get recrystallized into the capsule, then it is insoluble. Then rupture time of the capsule. So for soft gelatin capsules, rupture time will be there, and that rupture time will affect the drug release from the capsule. Then coming to the dissolution method development based on the CBAs. So we have identified the CBAs of the formulation and the API. Now based on these critical bioavailability attributes, a specific discriminatory dissolution method is required to be developed. So paddle method selected based on the formulation type of soft gelatin capsule. Also the paddle apparatus is very commonly used for soft gelatin formulations. Also paddle apparatus is used for rupture time determination. 0.1 normal SCl selected as the starting media, as Tmax is around 1 hour and the capsule gets ruptured in the stomach environment. So the 0.1 normal SCl selected as a media, so that it should have some biorelevance. Then surfactant and enzyme, pepsin and its level selected based on the formulation properties and the drug release. Now as the media is 0.1 normal SCl and it is in the acidic side, so this pepsin is used as an enzyme to digest the cross-linked gelatin. Surfactant is selected based on the API solubility and its concentration is justified. Then checking the robustness of the dissolution method. So to develop a dissolution method and to check the robustness of the dissolution method, changes are required into the SCl concentration, and that will have an impact on pH. pH of the dissolution media is required to be checked and studied in detail, and its impact on the release is is to be studied. Then change in the surfactant level in the dissolution media and change in the paddle speed. So robustness is checked by changing the SCl concentration, surfactant level, or sometimes the pepsin level is also checked, and the change in the paddle speed, so that hydrodynamics will be studied. Then the discriminatory power of the dissolution method is required to be tested or checked for the developed dissolution method. So for cross-linking CBA, that is critical bioavailability attribute of cross-linking, the forced cross-linking was done by the exposure to formaldehyde to study the effect of pepsin and its concentration required for the drug release and to differentiate the formulations. So this is for the cross-linking. Then we have identified the CBA like crystalline API presence. So use the capsule filled with the crystalline API which is insoluble at various concentrations like 0%, 5%, 10%, 15% and 20%. So see here 0% is the target, and if we vary the presence of this crystalline API, then it should reflect the dissolution. Crystalline API is not soluble, thus dissolution must detect the presence of crystalline API. It affects Tmax and the Cmax. If there is a crystalline API, then it will not get dissolved into the GIT and also into the dissolution media, and that's why it should reflect in the dissolution release. Now when there is a 100% API is present in the solubilized form, that time the dissolution shows 96% release into 15 minutes, and that continues to the 100%. If 5% crystalline form is there, it is coming around 92%, and last is 95%, so that this 5% is reflected here. Then 10% is also reflected at the last time point, or it is starting from the 15 minutes. 15% is also reflected, 20% is getting reflected, and 100% is also getting reflected. So here you can see the batch which is BE batch or target batch with 0% crystalline API is showing the highest release with more than 85% in 15 minutes. So the specification of 80% Q can be given in 15 minutes. Now the rupture test and correlation between the rupture time and dissolution is required to be developed. See here for the soft gelatin capsules, rupture time is very important for the drug release. Prior to the development of the dissolution method, capsule rupture testing is required to be performed during the stability studies on the lab scale and also on the pilot scale batches. The pilot scale batches means on the EB batches, in the water and in the acid media. So here the dissolution media selected is the acid media. So two medias can be taken for rupture time determination. The dissolution method also to be developed, because not always the rupture time will give the exact picture of the dissolution. That's why dissolution method is also required to be developed. Both rupture time and dissolution methods needs to be compared by demonstrating that the performance of in and out of specification batches are correlated. That means if the batch fails into rupture time, it will be reflected into the dissolution, and if the batch pass in rupture time test or rupture test, it will pass into the dissolution also. But here for this formulation, if the capsule contains crystalline API, that time the rupture test will be passed for all the batches, but the dissolution test may get failed. So the acceptability of the dissolution method and acceptance criteria is important. Acceptability of the rupture test during drug product development is very important, and correlation between rupture test and dissolution is important. In the current case, the recrystallization of API or presence of the percent crystalline form in the capsule cannot be detected by rupture test. That's why dissolution method is must and developed. It is must for the development. Then, based on the understanding of the formulation, the hypothetical example we have taken to have the clear understanding. For the CBS may be CMAs, CPPs, storage condition or anything. So these are interrelated. See, these factors which affect the discriminatory power and the factors which affect the bioavailability are interrelated. USB apparatus 2 pedal is selected at a speed of 50 rpm. It is a standard condition. Volume is 900 ml. Temperature is 37 degree. This is the standard volume. Also 500 ml can be tried based on the development. Tire 1 will involve 0.1 normal HCl with 0.3% surfactant. Surfactant will be optimized level. Tire 2 0.1 normal with the similar surfactant level with pepsin. Pepsin is selected because the media is acidic. The specification is given based on the biobatch. Not less than 80% at 15 minutes based on the Tmax and the percent release of BE batch. And percent release of BE batch I have considered the 0% crystalline form present, that batch is considered. So it was showing 96%. So the criteria of minus 10% is applied, and 96 minus 10% it gives 86%. So it is very clear that not less than 80% Q can be given for 15 minutes. So this is regarding the discriminatory and clinically relevant dissolution test, and these dissolution, discrimination and clinical relevance is now going towards the patient centric quality products. So nowadays the regulatory authorities are requiring that these dissolution specifications should reflect the clinically relevant requirement, and that's why the patient centric quality products are required to be developed. So the discriminatory and clinically relevant dissolution specifications and testings are always asked in the interviews. I hope you might have got a good understanding of the discriminatory and clinically relevant dissolution. Thank you for watching the video. Please do like, share and subscribe to Pharma Learning in Depth channel. Thank you.